Good afternoon again. Time for the last talk of Biospain 2021, which is going to focus on the technology which allows us to edit the genes of living organisms, Crispr. This panel is going to be moderated by Melqui Calzado, Secretary General of Catalonia Bio and Health Tech, Spain, and participating in the discussion we have Javier Terriente, CEO of C-Clinics, Spain. Thank you. C-Clinics, better?
C-Clinics.
C-Clinics.
Yes.
Yes. You can say it now.
The thing is that I don't think I can say it well, so I'd rather not say it.
So I will say it.
Yeah.
C-Clinics.
Good, perfect.
Let's try like this. Graham Garvey, Head of Genome Engineering Research and Development at Millipore Sigma, USA, and Abencia Sanchez Mejías, CEO of Integra Therapeutics, Spain. Again, it's time to learn. Thank you very much.
Thanks. Thank you very much. First of all, I would like to ask if it's Graham on the screen or not yet. Probably yes. Maybe we can continue. OK. So good afternoon, everyone, and welcome to this exciting session regarding CRISPR. My name is Melqui Calzado. I am the General Secretary of Catalonia Bio and Health Tech. This is the Catalan Association of Biomedical Companies. We are right now over 200 companies, and we try to help them to be more competitive on what they do every day. And this on a special focus on innovation, on networking, and also in the entrepreneurship. First of all, I would like to thank ACEBio for choosing such an amazing topic for this panel and thinking of me to moderate the panel. The history of CRISPR, a new genome editing technique used around the world, began just 600 kilometers far from here, in Alicante. The Spanish researcher, Francis Mojica, in 2003, discovered an inherited immune system in lots of bacteria that allows them to edit somehow their genome and call it CRISPR. This discovery remained silent until 2012, when Emmanuel Charpentier and Jennifer Doudna demonstrate that CRISPR is a universal system to edit any genome. They received the Chemistry Nobel Prize in 2020 for this achievement. So we have a really interesting topic to discuss today. Probably all of you, or most of you, have heard about CRISPR in the recent years. But what is exactly CRISPR? It is a revolution on genome edition or just an incremental improvement? Which are the main advantages of this technique? What can we expect from it as patients? In the next 54 minutes, we're going to discuss and try to answer these and other questions. It is a real privilege to have in the panel three extraordinary and complementary profiles that will help us to reveal the secrets of CRISPR. First of all, Javier Terriente. Thank you very much, Javier, for being here.
Thank you.
Javier is a biochemist with a PhD in developmental genetics. He has more than 20 years of experience in academy and pharma industry. In 2013, he co-founded C-Clinics,
Yes.
a vibrant biotech that exploits zebrafish as a research model for performing drug discovery, chemical assessment, and understanding human disease. C-Clinics provides different services to third companies and academic groups, while it develops internal drug discovery programs. Recently, Javier co-founded C-Cardio Therapeutics, a spin-out from C-Clinics, focuses on the discovery and therapies to treat cardiovascular diseases. Per his role in both companies, Javier manages a growing scientific team, has directed several PhD theses, and published multiple research articles. In addition to these roles, he serves as Vice President of ACEBio, the organizer of this fantastic conference. Thank you very much, Javier, for being here.
Thank you.
We have also on the stage Abencia Sanchez Mejías. She is a molecular biologist by training. She did a PhD in human genetics at Hospital Universitario Virgen del Rocío in Sevilla. She did 2 postdocs trainings, one in USA at University of Miami, and another one in the National University of Singapore, both molecular oncology and RNA biology. 4 years ago, she came back to Barcelona and joined the group of translational synthetic biology directed by Marc Guay at University Pompeu Fabra. There, they have developed gene writing technology for advanced gene editing. Recently, she has incorporated Integra Therapeutics to bring this technology to the patients. Thank you very much, Aventia, for being here again.
Thank you.
And on the screen, on the other side of the Atlantic, we have Graham. Graham, welcome to the session. Graham is a biologist and biochemist with a PhD in protein crystallography at the University of Wisconsin, Illinois, and he did a postdoc in the same university. He has more than 10 years of experience in the industry. He spent 6 years at Monsanto, where he led the technical teams to develop the first Monsanto vegetable projects for gene editing opportunities with CRISPR. And he also spent 4 years at Bayer, leading teams of scientists at sites in the US focusing on gene editing. From January 2021, he is the head of genome engineering R&D at Millipore Sigma in the United States. Graham, thank you very much and welcome to this session. So, Javier, could you explain us, please, in just 5 minutes, your relationship with CRISPR?
Okay, so I met CRISPR when I was a young guy. So no, CRISPR is a technology that came to us as a game-changing technology. So I was working as a researcher, working with zebrafish, still in academy, and we were trying to generate new transgenic lines, to generate new knockouts, to generate different tools for our research that would allow us to understand what is the role of genes in different biological processes. And to do that, before 2013 or 14, it was just very difficult. I mean, it was like extremely difficult, extremely painful. You would spend most of your time in the laboratory just doing molecular biology, just to try to answer something in maybe 1 year. So basically, it was just a very painful job. And then it came, well, not only CRISPR, but it came several technologies that were like incrementally showing some benefit one over the another. First, it was like a thin finger nucleases, then it was TALENs, and then it appears CRISPR, that is basically you put two things together, you inject it into an embryo, and you have a mutant in no time. And the way to do that is super easy, it's super cheap, and from one day to another, you are spending 1 year to generate a mutant, and now you can generate 20 mutants in 1 year. And so it was basically a game-changing technology for everybody that was doing research. It is, I think, a game-changing technology for people that want to use CRISPR as a way to modify the human genome for curing some disease. And it has allowed us to validate and to find and to discover new therapeutic targets, to discover the role of genes in multiple different indications. It just has opened the door to understanding much better the human biology, human health, animal biology, animal health, plant biology, plant health. So it's just an amazing technology. And in my company, I would say that 60–70% of what we do in our company is basically through CRISPR somehow. It's either because we use CRISPR to generate disease models, or we use CRISPR to generate, to understand the role of genes, or I mean, it's just something that is so fundamental for our activity that I don't think we should call it, instead of clinics, something like CRISPR, actually. So yeah, it's a great technology.
Thank you, Javier. It seems like you are married with CRISPR right now.
Yeah.
Okay. Yeah. Next move to Graham. Graham, from the other side of the ocean, what's your story with CRISPR?
What's my story with CRISPR? I think I met CRISPR about the same time the rest of the world did, and it was quite enlightening to see how fast you could move with CRISPR. What really is surprising is that pace of research that's improved with CRISPR. I want to take a little bit of time to introduce what's going on with CRISPR at Millipore Sigma, which is the U.S. life science business of Merck KGaA. Are we able to share the slides? I think I have some slides up being shared.
Okay, thank you, Graham.
If not, then I'll just keep going here. Sorry. I had a couple slides prepared just to kind of help talk through, but I want to talk a little bit about CRISPR at Millipore Sigma. We've been in the gene editing business for 15 years. Our company has a 350-year history of technical leadership. We started off with arguably the first version of an RNA-guided editing tool called Targetron. But as happens with technology, the next wave of innovation came, quickly surpassed that one, was replaced with zinc fingers, and moved very quickly on to CRISPR when that published. We've developed a couple of research portfolios. The first has been focused on the discovery end, where we are looking to enable researchers to explore genome modulation, gene knock-in, gene knockouts. We're providing tools like proteins and guide RNAs for researchers to explore these early discoveries. What we're really excited about currently is that we are developing a therapeutic-focused set of tools. As CRISPR has moved and evolved from a molecular biology tool that allows researchers to move more quickly to a therapeutic tool, we recognize that the need for what that tool comes with has changed. It needs to come with better purity, better safety profile, from better specificity, and from a improved documentation as it moves into the clinic. We're introducing this new portfolio of therapeutic-focused tools. We're calling it Pure Edit, and it'll be coming soon, and we're really excited about the improvements that are coming. To circle back to your question about my introduction to CRISPR, one of the challenges in the ag space has been the lack of tools and the lack of traits for focusing on moving the needle for consumers. That was my first introduction, but once you get down small enough and you're focusing on the proteins and the tools, it doesn't matter if you're in the ag system or if you're in the therapeutic space. Those tools need to work as promised. They need to be specific. You need to minimize off targets. And that's really where I've been focused for several years now, is how to make the tools that enable those specific applications, be they therapeutics or be they in the agricultural space. So that's been my introduction to the technology, and a little plug for our new portfolio of products.
Thank you, Graham. And now switch to Avencia. What's your story? What about you?
So my contact with CRISPR first, as you mentioned before, I did my PhD in clinical genetics. This was 14 years ago, I must confess. And it was a very exciting moment to be in clinical genetics. We could do so many things for families suffering from genetic conditions. We were the first team that bring into life a survival sibling of a patient that was affected, and the sibling was born without disease and compatible with the brother. So it was a very exciting time to be in this field, but still we could do little for patients with already existing conditions. And I think gene therapy has been an undelivered promise for so many years, and only now we are starting to see some progress in this field. So when CRISPR came to life in 2012 as a proposed technology for doing gene editing with therapeutic purposes, this was a revolution in the field. And now, fasting forward almost 10 years, still we have to understand that what CRISPR does is to find and cut the DNA, and the editing is done by the cell, actually. So CRISPR is good on cutting and disrupting, and now we are seeing more real editing tools being fused to CRISPR, such as base editors and prime editors, that could really do the editing job without depending on the cell itself. So taking this into account and knowing that CRISPR has some limitations in the amount of DNA that we can edit with these tools, and if we want to insert large genes and full genes that the patient needs for the therapeutic outcome, then CRISPR become a bit limiting. So we wanted to overcome these limitations. If we want to introduce full genes, then we traditionally rely in viral vectors, and this comes with their own limitation as well. The insertion is done in a random manner, and we have seen some patients with consequences because of this random insertion. So we wanted to overcome using CRISPR, but still overcoming these limitations that we saw in the field of gene editing and gene therapy, and doing something that was meaningful for a broader spectrum of patients. In Integra Therapeutics, we created a platform for doing advanced gene editing and gene writing, being able to transfer a full gene. And we took the benefits of CRISPR, which is very efficient and very good at locating a single target in the genome. And then we fused this with transposases and integrases that has been already evolved by nature to do this transfer of a full gene into the genome of the patient. So we have created a prototype, and in the company we do technology development to really bring this technology to a broader spectrum of patients. And as Javier mentioned, this is a platform that can be used for the agricultural business, also for generating models of diseases. But our focus is in therapy, and this is why we are pushing the movement forward.
Perfect. We will go deeper in this in a few minutes. Thank you very much for these 3 brilliant presentations. Now let's start the real discussion of the panel. And we know different genome editing techniques, such as interference RNA or the genomic overexpression vectors. But Javier, could you explain us which are the real advantages of CRISPR?
Well, the real advantage of CRISPR from an experimental point of view is that first, you don't need to be an expert molecular biologist to work with it. So basically, you can buy all the reagents for a cheap price and start doing things or editing genes very easily. So that's already a great advantage over other type of technologies that might require some deeper expertise of working in a lab. Then, if you speak about the other ways of interfering with the role of a gene, CRISPR allows to go straight into the region of the genome that you want to modify. So that and it does that in a very efficient way. So you can basically go and understand well which are the regions of the gene, or even the regions of the regulatory part of the gene that are important for either gene expression or for gene function, and you can answer this question in a very easy manner. And that's something that is not achievable in such an easy way with other type of technologies. This said, CRISPR is the holy grail, but it's not the holy grail. I mean, there is still some limitations, and there are still some technologies that have a lot of advantages, and depending on the type of question that you have, it might be more useful to use other type of technologies. But what is true is that, and I think that goes also in along the work that Avencia and Graham are doing, there's a lot of advancements in the tools that we are using now, the researchers working with CRISPR. So CRISPR was, let's say, if we compare CRISPR now with what it was 8 years ago or 9 years ago, it was very rudimentary, let's say, even though that we are still using most of the original tools that we were using back then. But now the tool set has expanded, I wouldn't say exponentially, but it has definitely expanded towards the use of CRISPR in different applications. So now there are better ways to generate point mutations than by using the original CRISPR with base editors or with prime editing. Hopefully there will be better ways to integrate large portions of DNA, either by homologous recombination or non-homologous enjoining integration of DNA, or by the use of transposases together with CRISPR. So what I mean is that even though it might not be useful for everything, the basis of CRISPR might make possible that it will be useful for everything in the near future. So I would say that one of the great things that CRISPR is bringing is also the versatility, that it can provide. So you can join it with so many things and work around this space that it makes it a great tool to play around. So yeah, that's, I would say, the advantage of CRISPR.
Okay. Javier, I was listening to you, and it seems like CRISPR is perfect. I mean, it has all of advantages. So I would like to ask to Graham, do you think that CRISPR has any weakness or any limitation?
So that's a really hard question to answer because that's sort of asking me to point out the flaws in a Nobel-winning prize. But I think it's a really astute question. So a couple of things come to mind. Specificity. When we think about how CRISPR came to be, it's a bacterial immune system. And so it's been evolved to provide that protection. The application that the cell is using it for, the bacteria is using it for, is to protect itself. And so some off-target activity could be advantageous to protecting it from mutations by the virus. That's one limitation, and that's why there's been a lot of research effort at academia as well as in industry to try and develop very specific variants of Cas9. The second area that comes to mind would be delivery. Avencia touched on this, that other molecules are being developed and the methods are relying on some traditional gene therapy methods such as viral delivery. But there's a limit to what you can package into the genome of the virus. And so CRISPR-Cas9 is a very large protein to begin with. And as we develop these other approaches such as base editing and prime editing, we're building those machines larger. And so there will be an opportunity to find smaller chassis, if you will, or frames upon which to build these editing machines. There's been a lot of effort already to find those smaller Cas proteins, as well as recent publications about how to package them, say, into a new virus delivery system as a protein. That came out of the Doudna lab earlier this year and was really exciting to see. And Feng Zhang's lab just published on omegas, which are transposon-derived RNA-guided nucleases. And so there's a large effort to find smaller proteins to overcome the delivery challenges. And the third area that comes to mind is immunogenicity. CRISPR-Cas has an ability to elicit an immune response. So when we're thinking about therapies that have a low-dose treatment where you'll need to go back in and treat the patient several times, that could end up being a challenge. And so groups have started to think about this, and there's approaches thinking about, when you deliver as an RMP, you can hold the cells long enough to allow them to cure, if you will, or clear the Cas9, and then be able to reinsert the cells back into the patient. But these are three areas that I think are going to be a focus of research in the years to come for CRISPR. They are the chinks in the armor of CRISPR-Cas. It is a great molecule that can do a great many things in a simpler and faster way. But these are the areas of focus when you think about the limitations of CRISPR.
Okay, thank you. We have been talking in these minutes on advantages, limitations of this promising technique. But now I would like to change the focus and try to talk on applications. So in 2020, a promising clinical study with 3 patients was published in Science magazine. So Avencia, CRISPR is a very recent technology. Do we have any more human results?
So yes, I have to say that some of these companies, Editas Medicine, Intellia, CRISPR Therapeutics, were founded in 2013. And only recently the first clinical trials were approved. So this has been a very anticipated results, and we were all really looking forward to see how applicable these techniques could be, if there is indeed any dangers due to the off-target activity, immune response in the patients. Also in the last few years, we have seen some results on traditional viral vectors in patients that were worrisome with AAVs. being not well tolerated in high doses, and some of the clinical trials having really adverse effects, and with Astellas having some patients that died because of the vectors. Also with lentivirus, we have seen patients that develop leukemias and lymphomas, and Strimbelis had to be discontinued for using in the patients. So we were really looking forward to see what would happen with CRISPR. And I think the most well-documented results were released this summer by Intellia Therapeutics, and they were looking really promising. So I think they did two things very well, these people in Intellia. They selected a disease that could be treated with the best thing that we can think of CRISPR. It was a hit and destroy target. It was a dominant protein that was accumulating in the blood and in the brain. So you just need to target this with CRISPR and cut, destroy. CRISPR can do this very well. And then the second thing that they did really well was to choose the delivery system. And I agree with Graham, the size of CRISPR is a disadvantage. It's a quite bulky protein. So what they did was to formulate this in a very similar formulation that most of us has undertake with the COVID vaccine. It was a lipid nanoparticle with a messenger RNA molecule that was targeting, in this case, systemically the liver, which is a well-validated delivery system, a big tissue that undertake most of the things that we could inject in a patient. So they obtained really great results with the efficiencies of up to 90% of editing in liver cells, and a decrease on 10-folds in the presence of this protein in the bloodstream. That was the protein that the patients were accumulating. And regarding the off-target, that was the big question out there. Could we treat patients with this in vivo that we cannot control? What is going to happen with the off-target? The outcome of this study reported very mild adverse effect, grade 1, with no detectable off-target that was significant. So this is very promising. And then regarding the immunicity, by having a mRNA molecule that we have tested in our system, you cannot find this in the body 48 hours after the delivery. So it's really a hit-and-run approach, and this was very promising. There is some other reports that are not that much based in science, with no clinical trial approved, and I think we all know about this, with this Chinese doctor that ended up in jail because he was editing primary cells in humans. But what we learned is that probably for primary cells, we don't have the technique as efficient as it could be. So the efficiency in this thing that he did was not 100%. And there is no off-target activity that was worth mentioning. And we have some pioneers in the use of these techniques that said that the off-target and the generation of mutation is probably close to what you get in a transoceanic flight. So I hope that we see more and more results in the next few years, and we can confirm this.
Fantastic. Thank you, Aventia. We will go back to the clinical applications in a few minutes, but now go again with the application on research. Javier, you mentioned before that CRISPR has changed in somehow how do you do research. I would like to ask to you, do you think that CRISPR is going to make a revolution in the way we do research? And how is this going to happen?
Well, I think CRISPR has already done a revolution in the way we do research. So it really has accelerated how we interrogate the role of genes in the context of disease, but also in the normal function in homeostasis in animals, in humans, or in animals that are models of how human biology works, but also in plants. But then there is another consideration. We are coming into an era now that is a deep tech era, no? where we have a lot of data, clinical data, omics data, etc. That is basically allowing us with the use of artificial intelligence. Yesterday there was a panel on artificial intelligence, with the use of artificial intelligence tools to really provide a lot of potential drug targets, a lot of potential mechanisms of action that might be involved in multiple diseases. You have to understand that I think there's only like 3% of the proteins are actually targets for drugs right now, of our proteins, which means that there's basically 97% of the proteome that could be target as a potential drug target. Obviously, not all the drug targets are pharmacologically targetable, and not all the genes make sense to target them by drugs. But still, what I want to say is that there's still a huge number of genes, together with a huge number of different diseases, which means that the intersection of these two missing pieces makes for, I wouldn't say infinite, but definitely quite a lot of things that we can still do, both in research and later in the clinical aspect, and that we don't know. And what CRISPR is allowing us, and I think is allowing many other people, is to be agile about validating this information. Okay, so what is allowing us to our clients, but also to us through the work that we do, is that you come up with a list of 50 potential genes that might be involved in obesity, and that means that when you mutate this gene, it might be involved in either promoting more obesity or less obesity. And when I say obesity, I can say Parkinson, or I can say Alzheimer, or I can say a cardiovascular disease. And by using CRISPR, well, I have to come and sell also my research model, and by using also zebrafish, but obviously other research models too, you can really go fast in interrogating these genes, in understanding if these genes actually have a role in the disease. And then obviously you can maybe use these models that you have generated to find new molecules. So it really helps to accelerate this virtuous cycle where you process a lot of information, you get a lot of useful potential information, and you can validate experimentally this information very fast.
Okay, excellent. Also, Javier and Avencia have the experience in human applications of this technology. And probably Graham is the proper panelist to talk us about the applications of this technique in agriculture. So what is this going to suppose to food, to agriculture system, this technology?
So one of the things that I'm excited about is this week there was an announcement that one of the first gene-edited foods is on sale in Japan. It's an edited tomato with a health effect for the GABA pathway. What I think we're seeing is in agriculture is very similar to what Javier just articulated. Is it's easier and it's earlier. 2012 was the first publication, and we already have products on the market that are going to consumers' hands, and they're being labeled as gene edited. So I think that speed of translating from a research tool to a product has been really amazing. The USDA has cleared quite a number of products for sale in the USA already, and they're not considering them to be regulated or different from standard methods of mutagenesis. I think this tool is allowing manipulation of genes that are very hard normally to find mutations from. You would have to go out in nature and search for a lot of diversity to find the gene that hopefully does what you want. With CRISPR, you can do a lot more input and design and control the outcomes. That ability to mutate the natural diversity and change it and accelerate that is really no different from what's been done for years with random mutagenesis techniques. So I think there's that opportunity to accelerate the rate of discovery in agriculture and meeting the needs for drought tolerance, for disease resistance, where nature just hasn't found the resistance gene yet, or the gene that allows it to adapt to that environment to keep pace with our changing world. So I think the ability to manipulate it quickly and screen through those mutations is really going to be where agriculture takes its best advantage of CRISPR.
Fantastic. Thank you, Graham. And now go back to the clinical application in humans. I made a search last week in clinicaltrials.gov, and it showed 22 different clinical ongoing trials based on CRISPR. So, Abencia, when do you think we will have the first CRISPR-based drug ready to use in our hospitals?
So I think we are pretty close to this point. These clinical trials that we have already the results of phase 1/2 that was sponsored by Intelia Regeneron with these really good results that were published in summer. They are already starting the phase 3. The week that this report was published, the stock value of the company went up 60%. So I think this speaks volumes of how soon we will be seeing the treatment. And on top of that, there are other clinical trials that are quite advanced in phase 1/2. One from Editas Medicine with inherited blindness and leberomaurosis, in which we will know the results next year. It's also looking promising, and then it will be a matter of finishing the phase 3. And there is another one with sickle cell anemia that already reported very positive results in this last summer, and is going to be finished the phase 1/2 next summer. So I think there are many more recruiting and approved, but these 3 are very close to be finished, especially the first one by Intelia. So I think it's a matter of a few years that we will see this being applied to patients.
Wow, amazing news. All of these 22 different clinical trials, all of them are performed in the USA or China. The question for all of you is Europe losing the CRISPR race?
So you mean the patients and the centers, or the companies that are, yeah. So yes, I think we have to do something better in Europe, and really it's almost a close monopoly in Cambridge Square, I will say.
Yeah.
So we really need to do things in this arena, both in therapeutics and beyond.
And is it a matter of companies or centers, or what do we need to change to move forward in this career?
So I think there are centers that are doing clinical trials in Europe. So I don't think that's the problem. Probably it's to really, and this is a very cost arena, and we really need to push entrepreneurship and really give the people the funds to bring this to the patients.
Okay. I don't know if Javier or Graham from the US wants to say something about this.
I think there's been some good discussion about the regulatory framework around this technology earlier. I'll comment on the ag side a little bit, and I think it's the regulations that are in place have been restricting to technology development. And I think it was called out earlier this week that some of the large companies have chosen not to move forward with some CRISPR development in Europe. So I think that's a place to look for the opportunity to change. On the therapeutic side, I think Avencia summed it up well, is those investments and those directions to promote it as a technology. I don't think it's a Europe-specific issue in that regard. I think globally we probably need to get a better framework in every country so that we understand how to move it forwards. And if there need to be new rules developed, then that's going to be the challenge, to put those in place to allow it to flourish.
Okay, so Javier.
Yeah, I just want to say that I don't think the difference is CRISPR. I think it's a global difference in terms of the intensity of the innovation between Europe and the States and China. I think CRISPR is just one of the areas where we have to push harder, but it's not the only one. I mean, we are I think if you look at the publication ratio in terms of high impact journals, if you look at the talent of our researchers, if you look even at the average research center in Europe, I mean, in Spain, in Holland, in UK, in Denmark, wherever you look in Europe in general, I think we have excellent research centers. We just don't have, I think, on one hand, we don't have the intensity of investment that you can find in the States. And probably we don't have the framework, and we don't have probably the mind frame to really push and to be entrepreneurial and to be ambitious about our research. So it's not a matter of CRISPR, it's a matter of having all this talent and making sure that this talent goes to the patients fast. And to go to the patients fast, you have to put the money and you have to believe in you. And that's what I think we are lacking more if we compare the European biotech sector compared with the States or even with China that is obviously pushed by the government. We are not being pushed by the government to grow. I mean, we should be pushed by the government to grow. We are not. We don't have enough investment neither from the private sector and neither from the public sector.
Okay. Graham, you mentioned during your intervention, you mentioned the regulation. So I would like to ask all of you, which are the regulatory challenges you are facing when you are developing these really new techniques?
I guess this is a question for me. I think the bigger challenge is how fast this field is evolving. And we still don't have a definite standard for measuring off target, for example. And this keeps moving forward with the techniques, and then the regulatory agencies catch up. So it's really uncertain when you plan to prepare for getting approved and what they will actually ask. And the time frames, especially with some of the results that we have from gene therapy and traditional viral vectors, probably the years that you will have to demonstrate that there is no adverse effect will increase with time, which is good for the patients and the safety, but sometimes we cannot afford having these very long clinical trials.
It's the same in the States, Graham.
Yeah, and I think the study from Intellia, they had really wonderful success, and I think it was, I'm blanking on which company it was, but when you do the ex vivo editing, what's the level of editing that you need to achieve before you put the cells back into the patient? And so are you going to put a regulation in place that is irrelevant to a new therapy, and then you have to go over this bar? So just think of it as if you have to achieve 80% of your cells having an edit, and those corrected cells then can go back in. But if the therapy was effective at 50% edited, you would still want to move forwards with it. So I think the regulatory framework is going to have to be flexible and be focused on the effect rather than a metric. And that might be a challenge into a field where we're learning so much quickly. Right? I think Avencia talked about the off-target. How much is too much? And what's the ratio where you'll take that risk because the outcome for the patient is so severe that that higher off-target might be worth it? Yeah. So I think that there's a lot of factors to consider as you move forwards, and how you set up your regulatory framework to enable addressing them on that individual basis and allowing flexibility is going to be critical, and not having a blanket, it's CRISPR, so therefore it has to be this metric of success.
Okay, so now let me make a futuristic question. Maybe for you, Javier. What other scientific uses might CRISPR have beyond genome editing?
Beyond genome editing?
Maybe Avencia or Graham, eh?
Yeah, I think the first CRISPR product was a COVID test.
Right.
So,
yeah, diagnosis for
diagnosis.
Sure. Yeah. Yeah, beyond genome editing, I would say that probably something that is not genome editing, but like mRNA editing, something that might not have to be permanent, but might affect the function of genes nonetheless. That's probably something that will be not so futuristic. So there are already some tools there that allow to modify RNA without having to modify the genome. And that might have probably an impact on both the research, but also on how we do gene therapy. In this case, it wouldn't be gene therapy, it would be like RNA therapy. Besides that, I think it's how to use CRISPR rather than other uses of CRISPR. I think basically CRISPR by itself as a genome editing tool, but also all the different set of other tools that are coming from CRISPR will help in many different areas, both from the research and from the therapeutic point of view. And once you have the tools, the possibilities are endless, right? So you can think about not only therapies for curing a disease, but maybe therapies that might make you more intelligent, or maybe, and that's also the ethical concern, right? Maybe it makes you more white. So that's the issue here that we have to face. When you have a tool that allows to modify the genome, do you want to modify the genome to make humans better? And what is better in a human? Is being taller? Is being a male instead of a female, or a female instead of a male? It's being more black or more white? It's being homosexual or heterosexual? So at the end, what CRISPR will make, what is funny is that we will have to come back to philosophy to really take down CRISPR and really make CRISPR an application that is helpful for all the humanity, not only for a few, and to make sure that CRISPR is an ethical tool and is not promoting inequality and human inequality, right? The rich people being better, the poor people being worse.
Yeah, that's
Well,
a good
it's
point.
amazing. Maybe I can suggest a panel for the next Biospain for ethical issues regarding CRISPR.
Right.
Graham, maybe some futuristic application, but in plants. Can you imagine or say something?
One of the things that I like about CRISPR, and it's part of the whole CRISPR system, is that it incorporates virus into the CRISPR cassette. And so a futuristic application could be in-field monitoring with CRISPR sentinels, where they're out there in the field and that capture mechanism is upregulated or overexpressed in such a way that you can start to really get an in-field survey of what viruses are now affecting your population and really get a real-time, within season readout for what viruses are challenging a specific field. And that would influence a farmer's decision about varieties. It might influence a research company's decisions about what to go target and look at. I think one of the most exciting things about CRISPR in general is that how new it is. There's really a willingness to embrace ideas about the tool and where to use it in a new space. I read a paper about a year ago on a really great application where they were looking at how CRISPR could accelerate enzyme catalysis, potential use in fermentation or in bioreactors. And so I thought that was a really great application of a tool where it's just one component of CRISPR. It's its ability to position things. That was a great publication, and I think it's those adjacent spaces where CRISPR-Cas is really going to find uses that we're not thinking of now because it's so focused on gene editing, but those adjacent spaces have a lot of opportunity, and those diagnostic kits are a good example of those products that can be developed that aren't necessarily editing the genome, but taking advantage of the capabilities of this class of molecule.
Thank you, Graham. Okay, now is the time for the question and answer for the audience. Both you are here in Pamplona and also the people that are seeing us from home. Maybe if any of you have a question, can raise your hand and we will answer you, both from home. I don't know if the backstage people will say yes if there is any question. The students in the here can ask as well. No? I know that this is not the better hour for a, for a conference like this, but I think it has been really interesting. So if there is no questions, just to sum up, we have talked about CRISPR, the advantages of them, have a lot of advantages, this new technique, but some limitations that we are trying to solve. It is a revolution on the way we do research. Javier has explained us this really deeper, and is really promising results in clinical trials that Aventia has explained to us, and she said that in a few years probably we will have this first drug based on CRISPR in our hospitals. This is really, really exciting news. I hope all of you have enjoyed and learned something about CRISPR in this session. Thank you very much, Javier, Aventia, Graham, for your time, for your participation. It has been a pleasure to moderate this panel. See you soon and enjoy Biospain. Bye-bye.
Thank you so much.